Okay. Okay. So I just want to make a couple more comments on the icosahedral symmetry. But are you happy with it? Are there any questions? And by the way, if I mention the number of hexons on a virus, don't bother to learn those. It's just so that I can say something. Okay, I was just going to show another movie, if the player will cooperate. I showed you herpes. Herpes is a very big virus, has a big capsid. The coronaviruses, which include polio, rhinoviruses, which cause the common cold, hepatitis A virus, they have very small capsids. As I say, pico means small. So they have very small capsids, so you can't look at them in an electron microgram. They are looked at in by X-ray crystallography. X-ray crystallography, though, has got the same concept that I talked about when I showed you that herpes virus picture, and that is because you look at things in crystals and you only see something which is in the same place in every virus capsid in the crystal. So again, you only see things that are fixed in space. If things are flexible and movable, you don't see them. So here we have a rhinovirus. Whoops. Well, this one isn't going to be very... Let me just see if I can play it. See if we can get this to be cooperative. Okay. If you look at this, let me just lower the lights a bit more. Actually, very dark. And now you can see the football glow. How about this, folks? This is and you'll see it better when I let the movie go again, but this is basically a capsid, protein capsid for a coronavirus, a rhinovirus in this case, which has had a bite taken out of it here. And one thing is you can see a penton here, this blue thing with clear five-fold symmetry. So that's one of its pentons. And on the face, it's a sort of fairly complicated hexon made up of individual proteins. And so that's the green and the red. The reason I'm trying to show this is where this bite is taken out, here, when you look on this surface here, all you see is blue or green or red. There's actually a protein on the inside, which is this yellow protein, which it plays a very important part in infection. And it lies underneath this blue protein that's on the outside. And really, the reason I'm showing it you here is that the inside of the shell can look very different from the outside of the shell. It really is a solid shell. It's not just a sort of faint lattice work. So this protein is really hidden from the surface. It's just visible on the inside because this is really a thick, solid shell. And I think it's probably easier to see that with the X-ray crystallography. So I'm just going to try and get this to play gently. Okay, but it's a bit difficult because I can't control the speed on this player. But I think you can see, just do it once more. So here again, you can see that hole in the center and this yellowy protein here. It's just not visible here. So these capsids are solid structures. They really protect the nucleic acid very well. Okay. Well, okay, and let's see if we can do it this way. Good. Okay, so that's the principle of the icosahedral nucleic acid, very commonly used by a lot of viruses. The other kind of symmetry that we standardly see, besides the icosahedral, is helical symmetry, so I just want to go over that briefly. And here is the classic virus that was used to look at it, which is tobacco mosaic virus, which infects tomatoes and things like that, but not us, and tobacco. But it still remains. The principles of the structure are the same. And it's built here of a nucleic acid which has got a helical symmetry down inside here that you can't see, and a series of capsid proteins that build up and are involved in its assembly. And my attempt to show that consists of a pair of shoes and some tubing. So the tubing is the nucleic acid. And what happens here is the nucleic acid is bound by these molecules of capsid protein. So this is known as a nucleocapsid protein. And so to form the nucleic capsid protein binds to the nucleic acid. Non-covalent interactions, but strong interactions nonetheless. And then another subunit will come on here. And now we've got two. And if the med school would buy me more shoes, we could have a nice virus. But as this goes round, this will build up into a nucleocapsid. Do you get the principle? Okay? Oh, you're brilliant. I was just checking I'd recorded that. So the length of this nucleic capsid is controlled by the length of the nucleic acid. Once it's got no nucleic acid to bind, that's the end of things. So the size in this case is just controlled by how many of these subunits you pick up. Now, if you've got imagination, and if I was allowed to go out and buy all sorts of shoes on my med school budget, I think you might see that if the heels were narrower and the toes were broader, we'd get a different diameter of our nucleocapsid than if we had narrow heels. So if I had big shoes like that, then it would cause a different diameter. So the size of these and the shape of them affects how tightly this is coiled. And the other thing is, if I had enough for 2 rows, if the shoes on top had, say, got Velcro on and could bind to the shoes underneath, I get a very tight, stable nucleocapsid. If they don't have Velcro on and I tossed it on the floor, it could probably just sort of unravel. And that's one of the principles that changes why these look different when you see them in the electron micrographs. Here I have a slinky. In this, I'm imagining that inside this slinky, you can only see the plastic nucleocapsid protein on the outside because it's protecting the nucleic acid that's inside that you can't see. But there's a fairly strong interaction between the different rounds of the helix. So when you see it, you tend to see it as a well-organized, very obvious helix. However, if I got less interaction between the rounds, well, there we go. If I had less interaction between the rounds, which is a flash cord that I stole from somebody who doesn't know, inside the flash cord is a metal wire, just like a telephone cord. So if this is your telephone cord, there's very poor interaction between each turn of here, so it looks very floppy, it's very flexible. So when we look at that, and we just throw it down on this, we get something that looks a real mess. But it's still in it's still helical nucleus symmetry. So the point is that helical symmetry, when you look at it in the electron micrograph, can look really pretty, like this slinky, or it can look just like a jumble, like the telephone cord. But they're both helical symmetry. So, as I say, it's the underlying principles that are important, and in neither of these can you see the nucleic acid, because in both it's protected by that nucleocapsid protein. Okay. So the helix may be stiff or flexible, and that really changes what it looks like when you look at these viruses in the electron micrographs. Okay, we've had that. Any questions on helical symmetry? Good. And then there's the hand waving. Pox viruses, I said you keep on coming across the true pox viruses. Pox viruses don't have obvious helical symmetry. They don't have obvious icosahedral symmetry. But they appear to have obviously some kind of symmetry. And again, when you assemble something from basic building blocks, there has to be some kind of symmetry in there. But it's not clear exactly what it is. And rather than give it a name, we call it complex, which basically means we've no idea what we're talking about. The pox virus, but all the other viruses fit very nicely into one or other of those. And then, as I say, the viruses can get an envelope, and that can be obtained by budding through a cellular membrane, except in the case of pox viruses, in which case it seems the membranes go to them rather than them going to the membranes, although that's still confused. But one thing about getting an envelope is if you get the envelope by pushing through the host cell plasma membrane, kind of like a virus going through a bubble of bubble gum. As it goes through, it gets surrounded by the bubble gum and it gets wrapped in it. So it gets wrapped in the plasma membrane. So it can get out of the cell, and if the plasma membrane seals behind it, it can get out of the cell without killing the cell. So one thing is that it enables the envelope viruses to just keep on getting out of the cell one or two at a time, and you don't know that they're getting out of the cell. The cell can manage to keep going if it hasn't done too much damage on other things, which it may well do. But some of these viruses, you can have them in the same cell for years, and the same cell can keep on releasing a few virus particles all through those years. So you can get a persistently infected cell that releases infectious viruses the whole time. Because the non-envelope viruses, they have difficulty getting out they get out by lysing the cell on the whole, so that the cell lyses and then they're released, in which case that cell's had it and it's not going to be able to release any more virus particles. So a lot of the viruses that cause chronic persistent infections turn out to be envelope ones because they can stay in the same cell. You can still get chronic infections from other viruses, but the mechanism is more different. So envelope viruses tend to persistent chronically infected cells tend to be associated much more with the envelope viruses, with the non-envelope viruses. And that's something to remember when you're studying for boards, because if you can remember one thing, you can probably remember the other half of it. So I said herpes viruses stay with you for life. The same cell can release them for a very long period. They're enveloped. If you can remember they're enveloped, you have an idea they might be chronic and vice versa. Similarly, when you come to the hepatitis viruses, hepatitis A and E tend not to cause chronic, or don't cause chronic hepatitis. B, C, and D do. And when you look at those, B, C, and D are the envelope ones. A and E are the non-envelope ones. And, okay, that's a little bit of things that you can learn for board. But why am I even thinking about this? Why is it so important? Well, one thing is chronic hepatitis is something that's clinically very important. But another thing is that I think I put a... No, I didn't. But the envelope viruses tend to be very much more susceptible to things that destroy lipid envelopes. Soap, alcohol, your gut. There are a few envelope viruses that have learned to protect the envelope and survive the gut, but by and large, envelope viruses don't survive the gut. So if we go back to those hepatitis viruses, A and E, the two non-envelope ones, are the ones you get by fecal-oral route because they can survive the gut. The envelope ones you get by the blood-borne route, direct contact to mucous surfaces, etc., because they don't survive the gut. So this has a big difference on how easy it is to inactivate the virus in the environment and how sensitive they are to simple methods of disinfection control versus non-envelope viruses, which tend to be much more stable. So rotaviruses, for example, which you can pass in daycare centres, which come out in what we call the poops, those are very stable, and if you toss diapers around and things like that, they aerosol, they survive on surfaces for ages. The envelope viruses tend to be much less stable, they don't survive on surfaces so well, and wiping those surfaces with something that disrupts envelopes can be very effective. And why is that for the envelope viruses? And I've already mentioned this, but the envelope viruses have to have an attachment protein. Since the attachment protein has to be on the outermost surface of the virus, it has to be on the envelope. So if you remove the envelope, you remove the attachment protein, and they can't even find their receptor, end of story. So if you could just remove the envelope from an envelope virus, it's a weak link. But fortunately, a lot of things like flu and herpes viruses, They're relatively unstable in the environment because they're enveloped, and they're relatively easy to get off by just wiping surfaces with an appropriate solution. So envelopes can get out of the cell without killing it, which means they can more readily establish persistent infected cells. They contain at least one virally coded protein because they have to have something to recognize the receptor. And if you lose the envelope, you lose the receptor, you lose the attachment protein, and the virus can't infect. So
is there a possibility that a non-enveloped virus can just become an enveloped virus by accidentally wrapping around the membrane? Or do you have certain viruses that are enveloped?
Okay. The question was, can a non-enveloped virus become an enveloped virus by getting wrapped in a cell membrane? It can get wrapped in the cell membrane. That can happen just by pure chance occasionally. But it's not going to be infectious because a non-enveloped virus will put its attachment protein in its capsid because that's what's going to hit the cell first. And it won't be in the membrane because if you remember back to your biochemistry, membrane proteins that are buried in the membrane are made in the Golgi and they go by the ER. They've got signal sequences to get them into the membrane and everything like that. So its attachment protein is made to be a part of a capsid structure. It won't have all those signals to get it into the membrane, to get it to the cell surface. So when it gets a bit of plasma membrane, it won't have an attachment protein in it, so it won't go anywhere. The attachment protein will be inside. So it might occasionally get an envelope just because the cell gobbles it in some way or other, but it's not going to be infectious. It has to have an attachment protein in the envelope, and that requires that it sends it to the right place. And that's something else. These viruses are all going to have to send everything to the right place, and that means they're going to parasitize your signaling machinery that makes sure that the right type of proteins go to the right place. And so, as I say, its capsid protein won't be made to parasitize the membrane protein signaling pathway to deliver it to the right place. It will go to the wrong place. But good question. Any other questions? Is there a question up there? Or? And this was just to remind me that the gut is pretty bad news for envelopes. As I say, a few envelope viruses can survive it, but very few. So if you know something is being transmitted fecally orally, the chances are very high that it's a non-envelope virus. So if you come across a few that are envelope that are doing it, memorize those. The rest are going to be non-envelope. So that leaves us with 5 basic types of viral structure. You can have a icosahedral nucleocapsid or a helical nucleocapsid. These can be enveloped or non-enveloped. And then you have Mr. Complex over here, the pox viruses. That's it. So all of those viruses that we had there fall into one of these groups. And in fact, when it comes to human viruses, so far we know of no so-called naked nucleocapsids, no non-enveloped nucleocapsids. Helical nucleocapsids, sorry. All the viruses that we know that are helical turn out to have an envelope, the ones that cause human disease. So, in fact, in terms of human disease, you just see these 4 groups: icosahedral, enveloped icosahedral, enveloped helical, or complex. So, all of those viruses that look so different on that first sheet come down to these. And as I say, the vast majority of them fit in these 3 groups as far as human disease is concerned. Any questions? Okay, now we have a couple of other small infectious agents that cause disease that I get the good fortune to talk about because Dr. Mayer couldn't find anybody else to talk about them, and they're not bacteria and they're certainly not the immune system, so I get them. And one of these are sort of what you might call, these are at the extreme ends of where you could think viruses could go. Viroids contain only nucleic acid, it's RNA, and they have a very small genome, and they don't code for a protein at all, so they don't have a capsid structure, and so far they're only known in plants. So why am I bothering to talk about something that's nucleic acid only, that only occurs in plants? And the reason that they appear here is because we have an infectious agent called hepatitis delta virus or hepatitis D virus or hepatitis delta agent because it's not a true virus. And so I called it agent here. And hepatitis delta is serious, and you'll come across it in your hepatitis lectures. So it's a serious complication of hepatitis if you get this virus as well as hepatitis B virus. And I included because it's got some features that are typical of viroids. Viroid RNA has some special features. And some viroids have got some very special features. And it turns out Hepadelta's delta agent is an RNA virus, much larger than true viroids. But its RNA has got these same special features in its secondary structure, and also it has some catalytic activity, which is unusual for RNAs. But some of the viroids have got these same special structure features, and they also have got enzymatic activity. So this has got very weird RNA. And it doesn't code for an attachment protein. It doesn't code for a capsid protein. It can't code itself. It's using it parasitizes. I don't have it here. But it parasitizes hepatitis B virus, and you'll be hearing about this later on, and wraps itself in the hepatitis B virus envelope. So it's actually parasitizing hepatitis B virus in order to get out of the cell. So here we have a virus of a virus, if you want to think of it that way, or a parasite of a parasite, and maybe I should ask Dr. Kafar to give these lectures. Anyway, this is another parasite. So it's a parasite on viruses, but it does have an impact on the nature of the hepatitis. So it's important clinically, but it's not really a true virus because it has no envelope of its own, has no attachment protein of its own. It assembles, as I say, using hepatitis B. So that's viroids. The other end of the scale, we've got prions, which are thought, although that's still a bit of controversy about it, but they're thought to consist of protein only. Some years ago, this had 3 question marks. A few years ago, I lowered it to 2 question marks, and now I got it down to 1 question mark. So we're progressing here, folks. But they're thought not to contain any nucleic acid, although there's some people who argue that it's just that we're too dumb to find it. But they're probably protein only, and I'll be talking to you about those in the last lecture of the virus section. And again, they're not true viruses. They have no nucleic acid. They just have the bad news. And it's very interesting. We'll come back to talk about those later. But that's okay. So those are the 2 extremes. You can also have agents that are only nucleic acid, agents that are only proteins, we think. This is just for the sake of tidying all these philosophical points up before we go into the viruses and their life cycles and their clinical aspects. Are viruses living or dead? I stand on both sides of that question. It depends on your definition of what's life and what's death. In cells, they do all sorts of complicated, elaborate things, and it's difficult to think of them as a pure chemical. Outside, they do virtually nothing, and it's difficult to think of them as really having any life to them. So it's just what you want to call them. It does affect some of the nomenclature a little bit because you will hear people talk about live viruses, and what they normally mean is if they say this is a dead virus, they mean if I inject it into you, you're not going to die. And if a live virus means if I inject it into you, you might get a disease. So live viruses and dead viruses are used, and if you say live virus, you think it can replicate. But very frequently, instead of talking about life or death, because how do you measure it when all you've got is a chemical there, basically, just protein and nucleic acid for the simplest ones, we tend to talk about what they do. So you'll hear viruses referred to by what they do, so many infectious units, so many lethal doses, so that you actually tell people how you're measuring whether this thing has got any capability to do any damage. I will be talking about that terminology later on, but part of that is because, as I say, determining whether the virus is live or dead is difficult. I mean, if you put a virus on the bench and leave it there, it's just going to sit there and nothing's going to happen. So it's it has a very boring life or a very interesting death, whichever way you want to look at it. Okay, what about classification? People have tried to classify viruses on all sorts of things until we really understood virus structure, and then we realized that actually the way viruses behave is much more related to their structure and their related viruses in terms of structure than it is in what they do. But there were attempts to group viruses on what they do because people thought at one stage that viruses did similar things would turn out to be similar. That's not the case. I've already mentioned hepatitis. Very different viruses can cause hepatitis. But you will find all the hepatitis viruses are lumped together because while you're talking about the impact on the liver, it's useful to refer to them all together because clinically they can present very similarly. So past schemes have included all sorts of things like the host range, the tissue infected, the type of cell, the mode of transmission. This is another one which was a useful grouping, and we'll have a lecture on them actually, called arboviruses, because arboviruses are the arthropod-borne viruses. And again, they include many different types of viruses with different types of life cycles. But as far as the public health people are concerned, they come together because these are the ones that you control by controlling insects, by advising people to wear long sleeves, use insect repellents, not go out when the insects are active, use screens, etc. So the way you control them from an epidemiological point of view is controlled by the nature of the vector, which means here arthropod-borne, mosquitoes, ticks, etc., rather than by the nature of the virus. So they are grouped together for sort of lecture purposes, for handling purposes. You'll find that people who work with the liver are familiar with all the hepatitis viruses, obviously. People who work with public health control are familiar with the arboviruses. And again, as I say, the methods are very similar in how you handle these. But if you really want to look at viruses and put them together based on a really useful classification scheme, it's turned out that the basic structure and the molecular biology are the ways to group them. So the classification, the official classification, is based on that. And viruses are grouped together by their nucleic acid, the nature of their capsid, whether or not they have an envelope, and their replication strategy. And I'll go through this one by one. So when you've got a nucleic acid, viruses can be RNA or DNA. And I hope by the time that you've heard the next 6 lectures, you will understand that whether or not your DNA or RNA has a big impact on how you go about replicating yourself. It's not immediately obvious, but actually if you think about cell biology and you work it out, the way that RNA viruses and DNA viruses can parasitize the cell is very different because the way that the cell handles DNA and RNA is very different. And we'll be going into that in later lectures. So basically, RNA viruses tend to have various properties which are rather typical of RNA viruses and not so typical of DNA viruses. So this does have a huge impact on how they behave and in some cases on the disease. Their genome can be segmented or non-segmented, and all that means is they can have one chromosome, in which case it's non-segmented, or multiple chromosomes. But in viruses, we don't talk about chromosomes, we talk about segments, again. Just the way that virologists developed, out of touch with everybody else. So a virus could have 8 chromosomes, but we would say it had 8 segments, whereas we can have however many chromosomes we have, but we don't say we have segments, we say we have chromosomes. These chromosomes can be linear, like our chromosomes, or they can be circular, like E. coli, for example. So again, those viruses are grouped according to whether they have a common type of structure with the DNA. The nucleic acid in viruses can in some cases be single-stranded, which again is different from bacteria and eukaryotic cells, which is always double-stranded. And so that again impacts the virus, and again I hope you'll see why as we go through these lectures. But if it's single-stranded, there's another thing to think about, because it could be the strand that corresponds with the message strand on the double-stranded DNA, or the strand that corresponds with the opposite strand. So basically, if you've got RNA, it could be RNA that can be translated as message because it's that sense, or it could be the opposite sense. And if it's by definition, if the genome could be translated because it's the right strand, it's called plus sense. And if it's complementary to the message, it's called minus sense. And we get both types of viruses. And again, as I hope you'll see, whether or not you package plus sense or minus strengths has a big difference on your life cycle and how you go about things. So the nucleic acid is sort of numero uno when you come to put these into different groups. And then you look at the capsid. And you look at its symmetry. Is it icosahedral? Is it helical? Is it complex? And that will very definitely sort it out into more families. Is it enveloped or non-enveloped? And we've already said how much the envelope impacts on the properties, and we've also said, these viruses breed true, as it were. You don't get viruses that were non-enveloped, and they don't have another family member that's enveloped, because that requires a whole different strategy. And you look at the number of capsimers. So I said, for example, that adenovirus has 12 hexons per face, 20 faces, 240 hexons, 12 pentons makes 252. All members of the adenovirus will have the same number of capsimers. All members of the herpes viruses have a different number of capsimers, but all members of the herpes virus will have the same as other herpes viruses. So the number of capsimers. But the number of capsimers, it will always be 12 pentons. It's the number of hexons that change. And to some extent you can see why, because if you've got a virus, the number of hexons basically determine the size. So if all members of the family tend to have roughly the same size genome, they're going to need the same size capsid, they're going to need the same number of capsimers. So these features will sort your viruses out into a series of families where all members of the same family have got the same properties, same nucleic acid, same type of capsid, same envelope or not envelope, same number of capsimers. And that was originally how they were all put into families. And that works very well, because if you tell me that you're working on a particular member of a particular family, I already know a lot about it, because once you understand the strategy of that family, you understand the strategy of the virus just from being told which family it's in. And we'll come to some instances where that's been really useful. But if you tell me you're working on some extremely rare herpes virus, I immediately know that it's enveloped, readily inactivated by various membrane active agents. I know it's got a DNA genome. I know that probably once you've got it for life. I know a lot of things about it without you even telling me anything more about the virus. I would be suspicious that your virus had all those properties. I would be certain it had an envelope. And once you've done all that classification, occasionally what we've found as we've come to understand viruses more is that the viruses that we originally thought had got all the same properties, as you start to look at what they do and how they do it, you discover they actually have different strategies about how they replicate themselves. And so sometimes these families are then subdivided, because as we understand more about them, we realize that they've been put together when they shouldn't have been, and we split those out. So the replication strategy, if we discover that things that were lumped together because morphologically they look the same, turn out to be somewhat different in terms of their replication strategy, we will then divide them. So the bacterial classification scheme has no particular evolutionary tree to it or anything like that. We just lump things together because it's useful. So I have given you a tree here. Don't learn the tree unless you find it useful, because you can find a lot of other trees where people have just arranged them in a different way. There's no real underlying necessity for this tree to be the way it is, but I've just done it so that you can get, if you want to refer back to the viruses we're looking at, you can use this as a quick aid to your memory. If you don't want to do, ignore it. So don't learn this structure as such. But in the DNA viruses, you can have double-stranded DNA, and the ones over this side, or single-stranded DNA, or pox viruses are actually double-stranded DNA. But then it can be enveloped or non-enveloped. And so we've got these divided down here. Herpes viruses, as I said, are enveloped. We've got the DNA can be circular or linear. So these non-enveloped adenoviruses have got linear DNA. Their chromosomes look like ours. They only have one. And these only have one as well. These are the papillomaviruses and the polyomaviruses. They both have got circular DNA. And in fact, they look very similar. They've got similar number of capsimers, similar DNA. They're both non-enveloped. They were formerly grouped together. We now know that they are actually very different, and so they've been split into two families. They were formerly in this other family. So this is just a way to organize them, and that's what it is. The classification scheme for viruses is just ways to organize them so they're useful. So this was my attempt to give you a useful organization. It's based on a textbook. Right. Here are the RNA viruses. Again, single strand positive sense, single strand negative sense, double strand. And so again, I've organized them for you. You can come up with other ways to organize these groups. It was just an attempt to do it. Any questions?
You said the pox virus was
Yeah, the question was, is pox virus double-stranded? Its genome is double-stranded DNA by and large, but it has a funny feature at the end. So that's why it was kind of off on one side. And I'm not going to bother you with needing to know that. But yeah, it's double-stranded DNA, the vast majority of it. Okay. So, any questions on classification? Okay. So, in these sort of general lectures, the next thing I want to do is we're going to discuss the life cycles of a group of various DNA viruses and versus various RNA viruses. But first I want to give you an overview of the general virus life cycle so that when we discuss the details, you've got some kind of overall picture to put it into. So the basic steps in the life cycle, there are 5 of them. Okay, 6. Absorption of the virus to the cell, so that's when it actually binds to the cell, and we'll go into each of these in more detail. Then penetration, because I showed you virus binding to a cell, it has to get into the cytoplasm in some way. So once it gets into the cell, that's called penetration. And then we've talked about how well armored these virus particles are to protect the family jewels from the environment. That's also going to protect the family jewels from being replicated or anything else. So you have to uncoat. Somebody once said to me, it's like the medieval knight. You know, he's got all this armor on and everything, and everybody has to carry him around. But once he wants to really do anything, he's got to take all that armor off before he can pass on his family jewels. So he needs to uncoat. You need to uncoat. Once the virus has uncoated, its attachment protein is no longer associated with its nucleic acid. And so you can't get infectious virus out of that cell. That's called the eclipse phase. It's there, but you can't see it. Just like when you've got an eclipse of the moon hasn't gone, you just can't see it. Then you make your new viral nucleic acids, your new viral proteins, you assemble them together and make a capsid. And in these larger viruses, there may be a maturation process because it's difficult to just assemble the capsid. There's frequently a maturation thing where the capsid is finally sort of screws are all tightened and it's pulled together. And you release the virus. And in a few viruses, you actually continue your maturation after release. We'll come to that later on. So those are the overall steps. And I'll just go into them in a little bit more stage. And then, as I say, for each individual virus, we'll discuss them in excruciating detail. Absorption. The virus comes, got an attachment protein on the outside of the envelope, if it's enveloped, on the outside of the capsid, if it's not enveloped. That is always a protein because it has to be coded for by the virus. And it comes in, and it will wobble around until it finds the cell surface. It doesn't say, hey, there's a cell, I'm going to swim over to it. So it diffuses around until it finds the cell surface. As I say, the receptor on the cell surface can be any component of the cell surface. But for each particular virus, it's a particular component. So a particular virus will bind to a particular receptor on the cell surface because that's what its attachment protein recognizes. And this will hold the virus in close contact with the cell surface so that now it can go ahead and penetration can take place. We'll go into that in a minute. But this process is basically just a question of diffusing till it meets the receptors, and then it's basically non-covalent interactions with the receptors that cause it to stick to the cell surface. So this is temperature independent. It requires that viral attachment protein because it's mediated by that protein recognizing the right receptors. And that's important for viruses. They don't want to bind to anything in the environment. Otherwise, they'd spend all their time binding to the mucus in your nose and they never actually manage to infect you. So they need to go to something that's going to be real. And we'll go into how they some of them make sure of that. But if they just bound to anything, they'd never find a cell. So they need to have something that tells them, this is a cell and I'm worthwhile infecting. And the cellular receptors can be anything. But temperature independent, it doesn't require ATP, it doesn't require energy, it's just an interaction. Protein something interaction.